Hi, and welcome to the third annual Seattle Cell Science Symposium. The goal of the symposium is really for all of us to meet each other. So we like as many participants out of the Institute as we can get so we can all find out what each other does and establish collaborations and do some cool science. So I'd like to say a few words. I'd like to start out by thanking our organizing committee, Caroline Hookway, if you'd stand up, Melissa Hendershot, Julie Cass, Tao Do, Caitlin Gerben, Winnie Leung, Angel Nelson, Jamie Sherman, Callista Yan, Teresa Bourne, and Megan Whiting. And if you weren't asked to speak, these are the people to talk to for next year. Not me. Okay. Thank you. Okay, I'd like to give an introduction to the Institute and what we're trying to do, but it's just an overview, and there'll be many talks and many posters and great opportunities to find out more about the Institute. So I think everybody knows that we're comprised of cells, and these cells have a variety of morphologies, and their morphologies are dictated because they're doing different things. So it's a classic case of structure-function relationship. And as we do more genomic work and more functional work, we find out that these simple categories of cells, these cell types, are becoming increasingly complex. We don't have good definitions of what they are yet, especially as the distinctions become more and more subtle. So we also don't know how cells interconvert and how we can get them to interconvert if they don't. So the lineages are not worked out very well. We also don't know how the genomes dictate what these structures are and what the functions are. Cells are not only exist in types, they exist in states. Cells can be in stress states, they can be in motile states, they can be in a number of states that we don't know about. This is much less characterized. One case is the cell cycle, where there's a number of states that are all seen. These we all know. We just look at the cells and we know what they are. Again, the structure of the cell tells you what the state is. For stuff like this, it's also true for other things, even though we may not have studied it yet. So we're interested in that. Third thing, so the questions that we want to do, or the issues we're interested in, is we'd like to create a high-dimensional cell atlas where we start to understand not only what different cell types are, but what their different states are, so we can see them and identify what a cell was doing, what it's doing now, and what it will do just by looking at the cell. And you'll see how we're going to do it in a second or two. Second thing we're interested in is how cells organize themselves. Cellular morphogenesis is really an unsolved problem. It's a fundamental problem. We don't know much about it. We're interested in that. Third thing is we'd like to know how cells transition from state to state. This is both a gene regulatory circuit problem, and it's also a morphogenesis issue. So clearly, imaging plays a big role in this, the way I've been spinning this. If our goal were met, we'd like to convert this world from a genomic world, where we're all in now, into a genomic and imaging world. So that's our goal, is to say that there's enormous information in images, maybe as much as there is in the genome itself. So the way we're doing this is we're trying to conjoin gene expression profiles of cells, the organelle distribution of cells, and the phenotype of the cell to make this high-dimensional atlas of states, and then study how they form and how they transition. And once we've got that sorted out, we then want to start to do a deep dive in a particular organelle, and the first one we're doing is the nucleus. We're doing this all in stem cells because they're just great for it. They're diploid, they grow well, their genomes are relatively stable, and they can be induced to differentiate into a number of different things. So they're just great for this kind of a project. So I'm not going to go through all this stuff. I just want to say that in doing our research, we're generating resources, and one of our goals is open science, is to share our resources with you. And I just want to have a brief overview of some of the resources that are available. One thing we're doing is we've GFP tagged all the major organelles of the cell, of the undifferentiated iPS cell and the cardiomyocyte, and they're all available. And they're QC'd very well. These are a great collection of cells. You can get them from Coriell. and on our website. Second thing we're doing is we're imaging cells, obviously, in a high throughput way or a medium throughput way. And we have a 4-channel imaging with bright field, the nucleus, the structure we're actually interested in, and the boundary of the cell for reasons that are not important now, but that's what we've got. And through that, we have a collection. We have thousands, tens of thousands of cells that you can mine, pull out mitotic cells, see that they're not all the same. It's amazing. None of these cells look the same. And see how poor our visualization tools are when we look at cells in 2D. They just don't tell us very much. So there's thousands of cells at allencell.org. We are creating resources on the segmentation front. To study these cells, we have to segment them and pull them out of the colonies in which they grow. Each of the structures, we have to somehow look at them in a detailed way and segment them to get their morphologies and where they are. And a toolkit has been created. You'll be hearing about this in one of the talks. This is an amazing toolkit that lets us segment cells in 3D in ways that we didn't imagine even last year. We also want to see things. We have a cinematographic rendering tool that's amazing. It takes these images that look like that, and they create them into something that really reveals so much information. And you can look here, you can see this is just astonishing, and the gap junction one's amazing. You can actually get the depth of the cell just from this type of a rendering. We're also doing label-free imaging, and I think most of you know about this, and the goal is, since we have thousands of cells, each of them with a labeled fluorescent structure, can we teach a computer to see the structure in the bright field images, which we also run? And the answer is yes, we can. It's quite spectacular, actually. Can't do all the structures, but we can do many of them. And we can run this on all the structures and then integrate them and create cells that look like this. And you see on the right there a mitotic cell, and on the left an interphase cell. So we can actually get to see not only where organelles are, but how they're positioned with respect to each other, all in the same cell. And we have other tools for doing this statistically. We also are beginning to conjoin our imaging with single-cell RNA-seq using FISH. You'll be hearing a talk about this. And there's posters on all this as well. And finally, we have our Allen Cell Explorer, which our goal is for not visualization to be figure 8, but to actually have visualization drive the science and help you think about things so you can do much better work. So we have that. All of our reagents, all of our data, all of our protocols, everything is on allencell.org. So with that, I'd like to thank our team. This is an amazing group of individuals. Everyone is spectacular and special. I've worked in a lot of environments, a lot of places. I've never encountered a group like this before. And I'd like to thank Paul, who loved the complexity of cells and wanted us to study it. So with that, I will hand it back to Caroline.